Hey friends, I am Jivish. I welcome to my channel Bioprofessional. In the previous lecture, I discussed the overview of cell signaling, and here in this video, I will discuss the Gpcr, that is the G Protein Coupled Receptor, and the signaling process. See how we communicate. We communicate basically by two means. Either we can communicate orally or we can write something for the communication. Just think, your body has trillions of cells, but they don't have any mouth or they don't have any pen or paper to write. So how they communicate? Because we know that cells make tissues, and tissues ultimately make organ, and every organ, whatever organ you have in your body, they communicate. They work in a very coordinated manner. If there is no cell-cell communication, or if the organs are not coordinated, they will not work properly. They will not function properly. So here in this lecture, we will understand that how the communication occurs. That's what we call it as cell signaling. Okay. So for cell signaling, we have to understand two things. At the very first, we have to understand what is called a receptor. Okay, what is called as receptor and the ligand. That is the very first concept you should have before starting of this cell signaling process. So here, see, suppose this is a cell, okay, it releases a signaling molecule. Okay, this is the very first step. That cell will synthesize a signaling molecule, and after that it will release that signaling molecule. Okay, so that signaling molecule undergoes for the transportation. So I will write it here. The very first point is that the synthesis of the signaling molecule. Okay, after that the release of signaling molecule. Number 3 point is that transportation, transport of that signaling molecule. Okay, and after that binding. Now question is that to which cell this ligand or the signaling molecule will bind. Okay, so this ligand will bind only to that cell which has got the receptor for it, Like if you go outside your home, you will most probably talk to a person whom you already know. The same analogy you can apply here. The ligand will bind to the receptor. But the receptor must be present on a cell. Only to that cell the ligand will bind. Okay. After that, binding means specific binding, and then the signaling will start. Signaling, right? We also call it as downstream signaling. Now, question is that we can classify the signaling process. Right? This signaling is extracellular signaling. Why extracellular? Because this signaling molecule comes out of the cell. It travels some distance, and then this ligand binds to other cell. So that's why it is called as extracellular. If the signaling occurs inside the cell, we call it as intracellular. Right? So we can classify the signaling process based on the distance traveled by the signaling molecule. Right? We can call it as autocrine signaling. Right? Autocrine signaling. Then paracrine signaling. Okay? And then endocrine signaling. So I am not going to describe all this process because I already discussed in my previous lecture. So if you have not watched the previous lecture, I will recommend you to watch the previous lecture and then watch this lecture so that you understand the concept very well. And I am sure that if you put your 100% attention, then you will of course understand the concept with very great detail, and you will be able to answer most of the questions that are asked in the CSIR, NET, GATE or ICR, whatever exam you consider. So please stay tuned and watch the complete lecture. Now first let me write the full form of Gpcr. This is known as G protein coupled receptor. Okay. So why this is called G protein coupled receptor, we will see, because it is associated with G protein. Okay. So what I wanted to draw here is that, let's say this is plasma membrane. Okay, we know about the plasma membrane. This is lipid bilayer. Okay, I'm not drawing like this. I just drawn two lines. Okay, to represent the plasma membrane. So G protein is basically a secondary structure of a polypeptide. Secondary structure. Now, secondary structure we can show like helix structure, right? Likewise, okay, this is actually multiple pass. And how many pass? This is basically 7 pass. See, 1, 2, 3, 4, 5, 6 and 7. This is 7 pass. So we are showing the structure of Gpcr. So we can call it as a transmembrane protein. What? A transmembrane protein. Transmembrane protein. Why it is called a transmembrane protein? We can also call it as integral membrane protein, right? Integral membrane protein. Why integral membrane protein? Because it has extracellular domain, right? It has extracellular domain. Okay, it has transmembrane domain. Here I am including here transmembrane domain, and it has intracellular domain. So any protein which has extracellular domain, transmembrane domain and intracellular domain, we can call it as integral membrane protein. Okay, here I am writing as integral membrane protein. Okay, integral. Now question is that this is basically a long chain of amino acid, right? This is the secondary structure of a polypeptide. Now question is that this is the amino terminal. This is the amino terminal, this is the carboxyl terminal. Okay, so we can write it here that amino terminal, N terminal is present on the extracellular side, extracellular phase, and carboxyl terminal is present on the cytosolic phase, right? Cytosolic phase. These are the properties of the Gpcr, G protein coupled receptor. Okay, we are discussing the receptor. Now another point, I am drawing here the same Gpcr. Okay, here I am now just simply making the structure like this, 7 pass. Okay, I can write it like this, 1, 2, 3, 4, 5, 6 and 7. Fine. Now Gpcr has got loops. It has loop like this and like this. Okay, it has loop. We can say that this is L1, L2, L3. It has here also, it has loop, 1 loop, 2 loop, 3 loop. It has loop structure. Means this part. So I think you are confused now. Let me just make you clear that, let's say this part is plasma membrane. Okay, so this structure is embedded like this. This structure is I am drawing the structure like this. Okay, and loop is present. This loop is present on the extracellular side as well as on the intracellular side, and this part is basically the part of plasma membrane. So here I drawn this part. Okay, so you can consider the plasma membrane like to that part. Okay, this much is plasma membrane, and after that certain portion is bulging out. This is also the part of the long chain of the amino acid, long chain of amino acid. This is the secondary structure of the protein, and it has loops. So now the important point is that it has largest loop known as L3 loop. L3 loop is the largest among all these, right? Now another point is that these three loops together make the ligand binding site. Okay, here the ligand binds. And this L3 domain or L3 loop, you can say L3 loop interacts with the G protein. We will see. Subsequently, we will see that L3 loop interacts with the G protein, and these three loops together make the ligand binding site on the extracellular phase. So now we discussed about the G protein coupled receptor, that is the Gpcr. Now we will talk about the G protein. Now question is that why it is called G protein? Why it is called G protein? Can we call any protein as G protein? No, we cannot call all proteins as G protein, because this G protein is named because it is associated with guanine nucleotide. It is associated with guanine nucleotide. That's why we call it as G protein. Now the G protein that is involved in the Gpcr and the signaling process, let us know the structure of this G protein. So this is basically a heterotrimeric G protein. Heterotrimeric G protein. Now hetero means different and trimeric means three different. So it has three different components. Okay, it has one alpha component. Let's say this is the alpha, this is beta, and this is gamma, right? So alpha component is the largest, and then beta, and then gamma. Gamma is the smallest one. Okay, so let us consider this is the plasma membrane. Okay, this alpha subunit is lipid anchored and gamma subunit is also lipid anchored. Okay, they are covalently joined here by lipid chain. So, and let's say we have here the Gpcr, right? Here we have Gpcr and here we have G protein, and this G protein is lipid anchored. Alpha and beta subunits are lipid anchored here, and the largest subunit is alpha subunit. Now let us look at the whole components together that is present in the signaling process. Okay, so we can draw here. Let us draw like this. I am showing, let's say, this is the receptor. Let's say I have drawn the receptor like this, and you know now it has 7 transmembrane helix and all, but I am not drawing here. Okay, and you have the, let's say you have, this is alpha subunit, this is beta subunit, and this is the gamma subunit. This alpha and gamma subunit is lipid anchored. Okay, and you have another component here present on the plasma membrane, right? Present on the plasma membrane. This is called the effector molecule. And this is basically the enzyme. We will see what is the enzyme here. So here now you have to just remember these components. It has one, this is the Gpcr, okay, this is the G protein, and this is the enzyme. Now what will happen? Now there is no ligand. Okay, there is no ligand. So if there is no ligand, that means this pathway is turned off. There is nothing going on. Okay, there is nothing going on. So now I think you are thinking that why this G protein is important. Okay, you will find that there are plenty of ligands present in your body, right? It is present on the yeast, on the plants, animals, everywhere. This signaling process is going on. Okay, so human genome encodes more than 800 different types of Gpcrs. Okay, almost 350 for for hormones, growth factors, and all, and for gustatory and olfactory. Gustatory means taste related, and olfactory means smell related. It has ligands, and human genome also has more than 100 Gpcrs, which are known as orphan receptors. Means their natural ligands are still not identified. So just now you can think that what's the plenty amount of Gpcr are present in the human genome and all the on plants and yeast cells also. So it has vast roles. Okay, it has many ligands like glucagon, it has calcitonin, parathyroid hormone, epinephrine. We will see there are plenty of hormones. These hormones are nothing but here the ligand. Okay, and we will see the ligands. There are many other different types of ligands are also there, and it will do, and it will ultimately do respond. Just I am telling all these things to you just make you realize that this process occurring continuously in your body. Let's say you don't have glucose level is shortage in your blood, and then what will happen? The glucagon will come and the response will occur, and ultimately the blood sugar level will be maintained. So how this process goes on? This is by the process of Gpcr, right? G protein coupled receptor signaling. So now we will take a general ligand. Okay, means a normal ligand. We'll not name this ligand, and we'll see what process goes on in the signaling process, and after that we will take one specific example and we will see what happens to that. So now please stay tuned and watch the complete lecture where I will discuss the ligand and the signaling process. So now let's understand the whole cell signaling process. So you can see here, this is the receptor and there is ligand. This ligand is present. Okay, now after binding to this ligand to the receptor, there is conformational change. Here you can see there is nothing shaped like this. So when the ligand binds to the receptor, there is a conformational change. So this conformational change leads to this G protein, right? Here I want to mention one thing, that when the G protein is bound to, let's say, this G protein when bounds to GDP, right? So when this is bound to GDP, it has higher affinity for, means G protein has three different subunit, as we know that it has G alpha, right? G beta, beta and gamma. So this when this G protein is bound to GDP, then there is higher affinity of this G alpha and gamma and beta. That means G alpha has higher affinity for beta and gamma subunit. Okay, and this condition we refer it as that G protein is inactive. Okay, G protein is inactive. But when the G protein is bound to GTP, right, its affinity decreases for the beta and gamma. That means there is a very less affinity. This G alpha has very less affinity for beta and gamma. Here affinity is more, here affinity is less. So, and in this condition we call it as G protein is active, or we sometimes call it as G protein is on. Okay, Now see, when there is the conformational change in the receptor, this receptor has, as we have already seen, that receptor has three lobes in the cytoplasmic surface. This is extracellular phase, this is cytoplasmic phase, okay, as this is the plasma membrane. So when the conformational change occurs in the receptor, means L3 loop of that receptor interacts with the G alpha, okay, G alpha. So this G alpha, and then the conformational change occurs in the G protein. So this conformational change leads to the replacement of GDP by GDP. Here you can see the G protein interacts with the receptor, and as a result this GDP is replaced by GDP. Okay. Now as this G protein is attached to GDP, now the affinity for the G protein, affinity for the G alpha bind to the, affinity for G alpha to beta and gamma is very less, so that this is now separated. Okay. Now this G alpha, this is G alpha subunit, now interacts with the effector molecule. So what is effector? As I already said, that effector is nothing but the membrane bound enzyme. This is not basically the membrane bound enzyme. Okay. Here in this case, this is adenylyl cyclase. This is what? Adenylyl cyclase. So this membrane bound enzyme, what it does, it actually causes the formation of cyclic AMP from ATP, right? Cyclic AMP from ATP. Now what's the role? So this is present in the cytoplasm now, cyclic AMP. Now what it will do? This cyclic AMP in turn activates one cytosolic enzyme. I am here writing that this particular cytosolic molecule, that is the cyclic AMP, activates one particular enzyme which is present in the cytosol known as pKa. This is protein kinase A. Now this is protein kinase A. As we are saying, this is kinase, right? That means it is an enzyme. It is an enzyme. So what is the enzyme nature? That means if it is enzyme, if it is kinase, that means it is an enzyme. It will phosphorylate some amino acid residue. So based on which amino acid residue it phosphorylates, we classify the enzyme, right? So what kinase? This is basically the serine threonine kinase, right? Serine threonine kinase. Okay. So, and we have likewise, we have histidine kinase. If the kinase phosphorylates the histidine residue, we call it as histidine kinase. If it phosphorylates tyrosine residue, we call it as tyrosine kinase. So here we call it as serine threonine kinase. Fine. So this pKa has two subunits. Okay. This pKa has two subunits. One is called a regulatory subunit. Okay, and two regulatory subunit and two catalytic subunit. We will see the detailed structure of that particular protein kinase A and how it gets activates. Okay, so for now you just remember these 2 points, that it has two regulatory subunit and two catalytic subunit. When this regulatory subunit binds to the catalytic subunit, it as a whole it remains in the inactive state. So for the activation, that catalytic subunits get separated from the regulatory subunits. So when this cyclic AMP causes the activation of pKa, so this catalytic subunit gets separated. Right, this is the catalytic subunit, this gets separated. This catalytic subunit is now active. Okay, what it does, it actually causes the phosphorylation of cytosolic protein like serine threonine, right, in the different position of a protein. Okay, polypeptide, it causes the phosphorylation. This is one part. It can also directly go to the nucleus. It can directly go to the nucleus and it phosphorylates one particular molecule known as CREB. What is CREB? This is nothing but cyclic AMP response element binding protein. Here I am, let me just erase this part so that I can write it here. So this is known as cyclic AMP, CREB, full form is cyclic AMP response element binding protein. Now when this CREB is phosphorylated by this catalytic subunit, it will further activate one particular transcription factor known as CBP. It will activate one particular transcription factor. This is transcription factor CBP. It is CREB binding protein. CBP full form is CREB, CREB, CREB binding protein. Okay, CREB binding protein. So this, as we know, the role of transcription factor. Transcription factor basically binds to the promoter region of different genes, and ultimately what it will do, it will activate some genes, right? Activate some genes, and we call it as downstream signaling. Downstream signaling. I think this is not shown here, so I am writing here. So it will cause the downstream signaling. So I am just briefly summarizing the part again, that when there is no ligand, there is no pathway going on. But when there is ligand, right? When there is ligand, there is conformational change in the receptor. This conformational change occurs in the receptor, and as a result, the L3 loop of the receptor interacts with the G alpha subunit, and after that there is conformational change in the G protein, and this G alpha gets separated from, means after that GTP is replaced by GTP, and then G alpha subunit interacts with the membrane bound enzyme, and as a result cyclic AMP is formed. And this cyclic AMP in turn activates one cytosolic protein known as pKa. This pKa ultimately phosphorylates some other cytosolic protein, or it can directly go to the nucleus and it activates the downstream signaling. Now, what you need to understand from here is that there is ligand, right? Ligand, we call it as primary messenger because it is carrying the message. Now, what is the requirement of this formation of cyclic AMP? See, cyclic AMP here, this is known as secondary messenger. This is known as what? Secondary messenger. So what is the requirement of that secondary messenger? Because if it is not present here, then this message cannot be passed to the cytosol. The message will remain on the plasma membrane, right? So for the delivery of the message in the cytosol and ultimately to the nucleus, and for the activation of different genes in response to that ligand, we require the secondary messenger. If there is no secondary messenger, this message can't be propagated into the cytosol and ultimately into the nucleus. So what you need to do is, now I will recommend you to take one pen and a paper and just write down all these steps I am writing over here and it will be very much helpful for you to revise the thing. Okay, So when there is ligand is present, let's say when ligand is present, okay, at the very first step there is ligand, and then there will be ligand receptor complex will be formed. This is the steps of the G protein signaling, right? So there is ligand receptor complex, and after that what will happen? Conformational change in the receptor. Conformational change in the receptor. Okay, there is conformational change in the receptor. Now the receptor has 3 loops. We know that now L3 loop of receptor interacts with G alpha subunit. Fine. Now the conformational change in the G protein. Conformational change in the G protein. These are the steps you can directly write on your notebook so that you can revise it in a very short period of time. So the first step is ligand, and then there will be ligand receptor complex will be formed, and after that there will be conformational change in the receptor. Here is the conformational change, you can see here, okay, and then this conformational change, that means the L3 loop of the receptor will interact with the G alpha subunit, right? This is the G alpha subunit, and after that conformational change in the G protein, right? Now GDP is replaced to GDP. Now GDP is replaced by GTP. Now activation of G protein. When it gets activated, the G alpha gets separated. G alpha separated from beta gamma subunit, right? G alpha is separated from beta gamma subunit, and then adenylyl cyclase becomes active. Adenylyl cyclase becomes active. Now adenylyl cyclase become active, and then it will form ultimately ATP to cyclic AMP. This is the step. So up to the formation of cyclic AMP, and after that we know that cyclic AMP in turn activate the protein kinase A, and this protein kinase A has 2 subunits. One is 2 regulatory subunit and 2 catalytic subunit. That's why protein kinase A is basically a heterotetramer. Okay, so this regulatory subunits, when it gets separated, the catalytic subunit becomes active and it can phosphorylate cytosolic protein on serine and threonine residues, because this is serine threonine kinase, and or it can directly enter to the nucleus and it will phosphorylate some other molecule, okay, and transcription factor known as Crab binding protein, so that it can induce the response. Here up to this part. In the next lecture we will discuss about the different secondary messengers. Here we have seen only one example of the membrane bound enzyme, that is the adenylyl cyclase. We have different enzyme called guanylyl cyclase, we have phospholipase C, and many other. So in the next lecture we will discuss all this part, and after that we have to discuss the termination part, because if a signal is there, means if a ligand is present and it is giving the response, then this response must be terminated. So every process must be regulated, and in a very coordinated manner we have to regulate every process. So we will see how this process is terminated. I think it will be very much helpful to solve many questions that already asked in the CSIR, ICR gate. Okay, because in every type of examination, like this type of competitive examinations, you have questions from this topic. So please try to solve those questions, and I will also try to solve most of the questions, so that you learn in a much easier way. So if you like my video, please subscribe to my channel, share with your friends, so that they also learn many new things and these concepts in a very easy way. So if you didn't write this information, then you can just take a screenshot and just save it to your mobile phone. Thank you.